In this video, we will discuss about this dendritic cells, antigen presented to the T cells, and the T cells killing of tumor cells. We will discuss how this process occurs, and we will not go through in the details, but we will just go through the interaction and how these dendritic cells helps in activating the T cells against the tumor cells. So it starts with this dendritic cells. Dendritic cells undergo phagocytosis by eating up the tumor cells. So at this point of time, the dendritic cells has the tumor antigen informations, and it needs to transfer this tumor antigen informations to the tumor cells. So first it will go through this lymphatic capillaries, through this ICAM interactions, and from that ICAM interactions from this lymphatic capillaries, it can move or translocate to this lymph node. In the lymph node, the T cells are also present. So here the dendritic cells interact with these T cells through immunological synapses, and thereby it will and here the interactions occurs through this ICAM-1 and this major histocompatibility complex 1. So this major histocompatibility complex provide the antigen informations to the T cells. At the same time, it will also provide the co-stimulatory signals to activate the T cells, which can be done by this ICAM-1 through these LFA1 interactions. And so now the T cells has the information about the tumor antigen, and it also gets triggered or activated by these ICAM-1 signals. So now T cells can produce the CD8 T cells, which is a cytotoxic T cells that has the antigen information against the tumors. And these T cells, through this blood flow, can enter to the tumor sites through this by interacting with this adhesion molecules such as ICAM-1, VCAM-1, ICAM-1, VCAM-1, or E-selectins also, and through that they can do this intravasculations at the tumor sites, and once it reaches to the tumor sites, it can undergo this immunological synapses with the tumor cells, and then trigger this cytotoxic effect through the release of granules, lytic granules such as granzyme, perforin. So that way it can trigger the tumor cell killings. So this dendritic cells phagocytosis not only kill the tumor cells directly, but indirectly also it will activate the other immune cells. So example here shown for the T cells activations. So if the tumor cells can prevent this phagocytosis through these immune checkpoints that we discussed previously, that one of the glycoimmuno checkpoint is the selectin, but there are non-glycoimmuno checkpoints also available, just PD1 and PDL1 interactions. So if those phagocytosis interactions are being prevented by the tumor cells, then it will not only affect the tumor killing by phagocytosis, but the other tumor killing by the other immune cells such as CD8 T cells will also get affected, as since the phagocytosis will not occur, so it will not provide the antigen informations, so it will inhibit the T cells activations against the tumor cells. So in order to improve this tumor efficacies, so what researchers has done is to prevent those do not eat me signals, or prevent the interactions between sorry, so to prevent the interactions between the checkpoint inhibitors on the tumors with this with their respective receptors on the immune cells. And some of the commercially available therapies are commonly known as immune checkpoint inhibitor therapy, or ICB, immune checkpoint blockade therapy, where they currently it is more popular for the non-glycoimmuno checkpoint blockade, such as this PD1 and PDL1 and the CTLA4. However, with the recent advancement in the research, people have found out that there are glycoimmuno checkpoint also available, such as CLEX7, 9, 15. So this can also be blocked to enhance the efficacies of the immunotherapies by not only activating the dendritic cells or macrophages against the tumors, but also helps in the proliferations of the CD8 plus T cells and the other immune cells. This is a brief overview about how this innate immune response works and how they activate the adaptive immune response and how they are correlated to each other. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.